Okay, guys, so photosynthesis chapter, I think this is like the fourth time I've started this, so forgive me if I zip through parts of it because I'm kind of annoyed. But anyway, here we go. So Bioflix, Pearson has a video out there. I couldn't download it embedded, it wasn't working for me, but they have what are called Bioflix. I think if you Google search them, you'll find them. But Bioflix, they have one on photosynthesis. It's helpful. It's not the end all be all, but it's helpful. It's worth your time if you want to take a peek at it. Definition of photosynthesis, we're going to convert solar energy into chemical energy. Who's going to do this? The plants. The plants and some of the protists, the algae, those guys. So technically three. Two big broad categories that we need to get are the autotrophs and the heterotrophs, and then a subcategory to the autotrophs are the photoautotrophs. So autotrophs sustain themselves without eating anything that they derive from any other organism. These guys are going to take in organic and inorganic molecules, things like water and carbon dioxide, and they're going to produce their own food. They're going to produce their own sugar. Photoautotrophs use sunlight to do that. They use the energy from the sunlight to make their organic molecule, and their byproducts are water and carbon dioxide. So again, we're talking about the plants, the algae, some of the protists, some of the prokaryotes, and they feed not only themselves, but pretty much the rest of the planet. So just some visuals to help you. Cyanobacteria, these guys are pretty cool. We didn't touch on them as much as I wanted to, but when we look at mitochondrial DNA, I know in chapter 6 we talked about mitochondria and we talked about how they have their own DNA and how you get your mitochondrial DNA from your mother. But when we trace back the DNA code on mitochondrial DNA, it comes back to cyanobacteria. So there's a really cool concept out there called endosymbiosis. Let me see if I can type that out for you. Let's see if I can do that. It's called, oops. Endosymbiosis. And endosymbiosis, I should actually have put that down here by the cyanobacteria. Sorry about that. But endosymbiosis is pretty cool because it it's where we talk about cyanobacteria and mitochondria and how we think that cyanobacteria either were ingested by early organisms or they were a parasite to that organism, and that's how their DNA ended up forming in mitochondria. But pretty cool. It's called endosymbiosis if you want to check it out. Heterotrophs, the counterpart to the autotrophs, get their organic material from other organisms. So they're the consumers. They eat stuff. They either eat plants, so they either eat the autotrophs, or they eat other heterotrophs. So they're going to eat somebody or something. Chloroplasts are the organelle that we're talking about. That's going to be our site of photosynthesis. And like I said, chloroplasts are really similar to some photosynthetic bacteria, specifically the cyanobacteria. And that term again is endosymbiosis if you want to check it out. So we've got some structures we've got to look at. We've got chlorophyll, that's the pigment inside the chloroplasts, right? Chlorophyll is going to be our light absorber. It's going to absorb a specific wavelength of light, and we'll talk about light here in a few slides. Carbon dioxide is going to enter the leaf, and oxygen is going to exit the leaf through some microscopic pores on the underside of the leaf called stomata. Don't confuse stomata and stroma. Make sure you're careful about that. I need to take a pause here because I think the dogs are about to bark, so hang on. I was right, barking happened. Okay, so we need to see the visual on this. We need to see the mesophyll, the thylakoids, and the grana. So I'm going to skip to the next slide. You guys can come back and or look at these, excuse me, or look at these on your PowerPoints. But here's what we're looking at here. So we've got a leaf, right? And if we zoom in here on the edge, what we're going to see is we're going to see the outer epithelial layer on both the top of the gray on the top here and the gray on the bottom. Okay, and then we've got this layer or two layers of cells in the middle, like Urolodia had two layers. Okay, so these are filled with mesophyll or mesophyll cells, and that's where we're going to find those chloroplasts. So here's a mesophyll cell, here's all those chloroplasts. Okay, so mesophills on the interior. Typically, a mesophyll cell is going to have between 30 and 40 chloroplasts in it. You saw a lot on Urolodio, and I think probably the best example you saw was when you were looking at cytoplasmic streaming or when you were looking at the salt water, the salt and the tap water concentration. Remember from chapter, I don't know, probably 6, we talked about each one of these little flat discs being called a thylakoid, and that the chlorophyll was within here, within these chloroplasts, within the thylakoid, okay? And that this was part of where we were going to do light absorption. As we put a whole stack of thylakoids together, we get a grana, right? So we had a individual was a thylakoid, a stack of them are grana, and that grana are interconnected. And then remember we had a liquid that flowed around and in between all of these granum, and that liquid was called stroma, S-T-R-O-M-A. Don't confuse stroma with stoma or stomata. Stoma or stomata are the holes in the underside of the leaf where we bring in carbon dioxide and release oxygen. Stroma is the liquid in between the grana. So don't mix those up. Bigger blown up version. Here you've got some matching, so go ahead and match those. I'm going to count to 5 and then give you the answers. And I'll count to 5 again. But you can always pause. Remember, you can pause video whenever you want. Okay, here is the chemical reaction for photosynthesis. It is a balanced equation. I need you to learn the balanced version. I don't want the generic carbon dioxide plus water plus light gives me sugar and some oxygen. I don't want that. That's high school level. I want you to start balancing this equation. I want you to know how many carbon dioxide, how many waters go in, and what we're getting out of it. And to help you with that a little bit, here is how we do that. So we're going to split up this water and we're going to import, or we're going to incorporate, I guess maybe, the electrons of the hydrogen into the sugar molecules. Okay. Part of the oxygen from the CO2 is going to come over here into water. All of the carbon that we get from the CO2 obviously is going to go into the sugar, and then a good chunk of the oxygen is, or rather all of the oxygen as well, is in sugar is going to come from that carbon dioxide. But just to help you understand where it's coming from and where it's going to, this is going to be my pause point. Nope, I'm going to do a couple more. So photosynthesis is a reduction oxidation process. So remember, reduction and oxidation had to do with electrons. Remember, if a molecule was reduced, it had gained electrons. And if a molecule was oxidized, it had lost electrons or had donated electrons. So when we talked about cellular respiration, we had 3 parts. We had glycolysis, we had citric acid cycle or Krebs, depending upon how you called it, and then we had oxidative phosphorylation, and then oxidative phosphorylation was made up of the electron transport chain and chemiosmosis. Remember that electron transport chain was huge in reduction and oxidation because we were moving those electrons from one protein, from one cytochrome protein to another cytochrome protein. Here, we've got reduction and oxidation as well. So this photosynthesis is also a redox process. We subdivide it into 2 parts. We call it the light reactions or the light dependent reactions, which is how I prefer to refer to them. And then we have the Calvin cycle or the light independent reactions. Some authors will refer to the events of Calvin cycle as the dark reactions, and I just don't like that. It leaves the implication that it has to be done in the dark, and that's actually not the case at all. So here's our overview. Remember our end goal for this plant. What this plant wants is this plant wants to make a sugar. It wants to make a sugar, okay? It wants to make a sugar, this guy right here, so it can take that sugar and send it to the mitochondria where we can do cellular respiration and make ATP. Okay, so I'll count to 5 and then I'll move on, but pause the video so you've got time to do this. Okay, this is my stopping point. We'll pick back up with light and light spectrum, but this is video 1 of photosynthesis.